Let me briefly remind you what we saw yesterday. So I showed you initially this experimental data by Bags and Plants from 2003, in which they measured local field potentials, and they observed these nice power laws in the size of the size distribution of these avalanches, with exponent 3/2, and exponent 2 for the duration of the avalanche. This is ugly, and it's always uglier for avalanche duration distributions. And what I showed you yesterday was that these kind of beam field directed percolation models, in which have a phase transition between an absorbing and an active state, had some pros. It could correctly predict those neuronal avalanche exponents. It could correctly predict these response exponents that you see in psychophysics, and some neurophysiological data. It predicted this dynamic range, then was later measured in some experimental setups, and therefore it provided some kind of useful picture, some theoretical workhorse of sorts for over a decade. So this was again the picture of the model. So I have a model in which for low coupling things go to silence, and then this is called an absorbing state. And if the coupling is strong enough, you have this self-sustained activity here in blue. And if this topology is sufficiently mixed up and messy, these exponents are 3/2 and 2, and this is not experimentally. By the way, yesterday I was mentioning this model. I forgot the name of a model that looks like this. I mentioned the benching process, but what I wanted to mention was the Galton-Watson model. So the Galton-Watson model was a model proposed in the late 19th century to explain why some family names in Britain survived, whereas others didn't. And guess what? Those who have more kids win. That's it, right? And so this is like a branching process. Here is a guy having lots of kids. Here's a guy having few kids. Then its family name disappears. What? More boys. Yeah, there's. Yes. Okay. Yeah. So just to remind you again. So we had this third exponent here for this universality class, which at the critical point is like 1/2 here, and it corresponds to what you see in psychophysical response, and some retinal ganglion cell response, and some detailed retinal model. I'm just repeating so we can refresh our memory and move on to the messy stuff of today. So there's this dynamic range prediction from the model, which it peaks here and it peaks in the more. I mean, it doesn't grow that much here in the experiment. Two different experiments. Preparing you for tomorrow and the day after tomorrow. Experiments in which this is a cortical slice, okay? And this is an anesthetized rat. Anyway, I'm talking about a functional advantage here in a model, which is nice. And here I'm talking about functional advantage for things which are not alive. I mean, this is not even an animal, and this is an anesthetized animal. But okay, right? Just to refresh your memory for things that I'm going to talk about on Friday. The original idea for this whole proposal of criticality for people who initially thought about it, well, presumably it gives you functional advantages. And right now I'm talking about slices and anesthetized animals. So I don't, right? Just to keep our expectations balanced. And, well, so okay, so it gave us some useful picture. By the way, optimal dynamic range, pretty much any model with an absorbing phase transition will have the maximum dynamic range at criticality. So in that sense, there's nothing special about this universality class. It's just a geometric thing, by the way, right? The way those curves behave. So it's pretty it's barely a result. Right, okay. So, great. So what happened? I was very happy with those results, and I was giving talks, and by that time, Siddhartha Ribeiro was leading the effort of building what is now called the Brain Institute in Natal, the northeast of Brazil, Federal University of Rio Grande do Norte. A magnificent endeavor which has grown. Now they have a beautiful building. If you can, just go visit because it's very nice. And he's a biologist, a neuroscientist, and I was welcomed there, and he was very generous. And I gave the talk, essentially the talk I gave you yesterday, and he said, "Well, okay, nice. You have this criticality idea stuff. So I have this bunch of data," and then he showed me a DVD. You guys know what a DVD is? You still know it? Yeah. So it's how you pass along data and music and movies. Well, he gave me a DVD. Here you have some data from real animals doing real stuff. Get me some criticality out of that, if this is such a good idea. And I am, I don't know, a theoretical physicist working on models with a bunch of students in my group, all of them doing models and simulations and whatnot and calculations, right? And this is data, I'm going to describe this data further in more detail. Data from non-anesthetized and freely behaving rats. You have these implants with maximum 32 electrodes. So again, Beck's implants experiment had 60 electrodes in a petri dish. This is the technology initially developed in Miguel Nicolelli's lab, in which have several tungsten electrodes, which you can implant in rats and then in monkeys. And you make a chronic implant. You fix it with dentist cement, and it's fixed there. The animal is okay. It recovers after some weeks, and then it can go. You can move around, etc. Right? And Siddhartha explained me it was all data for them from an older paper of theirs, and I go back home to my group meeting. And I hold the CD. And a bunch of students are sitting, my students, right? My PhD, master students, undergrad students, say, "Okay, guys, I've never done this before. I barely can do models and stuff like this. That's what we've been doing for some years. But I won't do this alone. I need a student to do this. Who would like to do this?" And then there was some silence. Like people take a step backward, right? What? She wasn't there. Okay, yeah, she was not. She wasn't even born yet. Yeah, right. So there was a silence, and I understood the silence. Just huge sociological parentheses here. In Brazil, there's this tradition of theoretical physics, sort of tradition, I mean, to some extent, and there's a tradition of doing some complex system physics in models, and nice, which is nice, nothing against it, but it took people in Brazil a long time to start dealing with real data. Okay, let's face the data and see whether, I mean, it's very easy to work on models and get excited about them, and get too over-enthusiastic about the models and then forgetting that at some point they should be compared with the reality. So my students were there. And then Tiago Ribeiro, they're not related, by the way. Ribeiro is just a very common surname in Brazil. Said, "I would like to do it." He was then a recent, I don't know, an undergrad or maybe starting his master thesis. Still without a project. I think he was among the youngest of my students. Today he's at the NIH. But Tiago is a very quiet person. He doesn't speak much. But whenever he does, something right comes out of his mouth. I don't know. He's one of these kinds of guys. And I'll do it. And then we started this. It took us a very long time to work on this data. It took his master's thesis, his PhD thesis. It took a long time for the paper to come out, and that's more of the story I'm going to tell you. So lots of pitfalls along the way, things we didn't understand, and I hope that while I'm telling the story, if you get confused, welcome to my world. That's how we were for a long time as well. I'm just sharing with you my ignorance and the things, the difficulties we had, so you can appreciate perhaps that things get messy. Right? So this is work done by Thiago Ribeiro mostly. And along the way, we needed help. So we got help from different people along the way. And with further experiments being done in Natal by Siddhartha's students in the Al Belchior, and then Fabio Cacheta. And then we had input from Dante Calvo, an Argentinian who works in criticality for a long time. He gave us very nice ideas. I'll comment on that. So the data Siddhartha gave me came from an older paper of theirs, which was designed for their own scientific agenda. That's why you'll notice it's so complicated. It's complicated for us, but for them, it was just their thing. So they were interested in learning how rats learn and how that learning depends on sleep. So Siddhartha is a sleep specialist. Okay? So these rats are freely behaving rats that cycle along the sleep-wake cycle ad libitum, as they wish. Okay? So they sleep and wake up and everything, and by that time they could characterize this very well. They know just by looking at the electrophysiological signals whether the animal is asleep, awake, slow wave sleep, or REM sleep. They can do it just with LFP signals. Right. So they implanted 3 brain regions: primary visual cortex, primary somatosensory cortex for the whiskers, because I will show in a minute, these animals were going to explore their cage and find novel objects, and they explore this thing with their whiskers mainly. And hippocampus, because hippocampus is involved, Bill mentioned yesterday, navigation, but hippocampus is also involved in memory formation more generally. So they wanted to look at that. And why V1? V1 was supposed to work as a kind of control area because the experiments were done in the dark, so nothing interesting should be happening in V1, right? And so they had the experimental protocol, which was interesting for them, in which the animals were in a cage to which they were accustomed. So they were there for, I don't know, days, weeks, or something. So it was uninteresting, just their home. And they're here, they wake up, they go to sleep, they dream, whatnot. And for 2 hours, they record this pre-exposure phase of the experiment. So the animals have been implanted long before that, okay? And then novel objects are introduced here, and the rats go crazy because there's novelty. So they explore it. Then there's this ball, the urchin, this thing which I forgot how it's called, and then there's some food that they can explore here. And then they go crazy, they explore this thing. And then they remove the objects and leave the animals here. And if you ask Siddhartha, he probably tell that he would bet the animals are dreaming about the objects or something, etc. And you keep measuring this thing. And they do all kinds of molecular biology experiments, very sophisticated things like sacrificing the animals in different phases here and showing the expression of different genes in different regions of the brain to see what is controlling memory formation and what is not, how this depends on sleep and what is. It's a very interesting paper, if you want to read it. Nothing to do with what I'm doing here, but this is the data that he had, right? So the fact that he had this gene expression signatures in their data is why this is called the GE series. So the animals are GE1, GE2, GE3, then we change their names later. And the data he gave me was essentially spikes. It's for two reasons. For computational reasons, it's way lighter, just the instance where different neurons spiked, and also because it is generally understood that a spike, well, is what the neuron sends out through its axon, whereas local field potentials, it's harder to model. It's like this average field around the electrode, which is generally believed to be caused by currents in the synaptic tree of the, so it's the input of the neurons, not the output necessarily. So some neuroscientists attach more value to spikes than local field potential, depends on their questions, right? Along the way, we found it necessary, because we were not understanding the results, I will share with you this puzzle. We found it would be interesting to have some anesthesia data to compare with. So eventually, Fabio Cacheta and Yael Belchior, Siddhartha students, did some extra experiments with the same kind of setup, the same strain of rats and the same kind of electrodes with ketamine, xylazine anesthesia to compare. For a long time, it was my first experiment, my first experience with experimental data. I just refer to those animals as anesthetized animals, as probably many of you physicists will do. That is anesthesia, right? And no, there's no such a thing as anesthesia. There's propofol anesthesia, ketamine, xylazine anesthesia, urethane anesthesia, etc. They are different beasts. They do different stuff. We don't fully understand what each of them does, and there's people who dedicate their lives to understanding that. Okay, so this is the ketamine, xylazine anesthesia. It's a kind of anesthetic. It's a combination, importantly, from which an animal can recover. You come back from this anesthesia. Okay? And the animals will in those experiments. We had about 7 rats in total. Okay, so just come back here for a minute. Please notice that we have 3 behavioral states which are classified in the data. We have 3 brain regions, and we have 3 phases of the experiment: pre-exposure, during exposure, and post-exposure to these novel objects, and therefore 3 times 3 times 3 is 27. Whenever Tiago knocked on my door to show me new results, he had 27 multiples of whatever we were looking at. Right? What I'm going to show you today is just a very well sifted and filtered version of what survived of 27 times attempts to understand what's going on. So we're looking at spikes, not local field potential. Let me show you what the data looks like. So in red here, it's just an example from one animal from data from the primary visual cortex. So each little dot, little line like this, is a spike. Each line corresponds to one neuron, which has been identified, like Bill mentioned yesterday, through spike sorting. So I have these electrodes. It picks up signals from neurons around it. Actually, differently from what Bill showed yesterday. It's a little bit different because this is older technology. So we have one electrode here, one electrode here. I forgot the distance here, but it's like 100, 150 microns apart. So if your neuron is here, it's probably going to be picked up by just one electrode. But this electrode will then pick up signals from different neurons, and then you have to sort this guy from this guy from this guy just by looking at the shape of the electric potential that you get from here, which is different from this one, which is further away, so it's weaker, something like this, right? So there's this whole spike sorting, which is different because it doesn't get picked up by two electrodes, probably. But anyway, you see here that slow wave sleep has this name for a reason, because you can see this periodic, not quite, but you have some characteristic time here, and there's local field potential, and in spikes, more or less. Waking activity is more asynchronous. It's kind of messy. You don't see, I hope, much structure there. It's not easy to see like this. And REM sleep, which is when you're not only, it's not the only occasion in which mammals dream, but it's typically when you dream, okay? So there's a whole debate about that, about dreaming and REM sleep. So it looks pretty much the same like this one. The electrophysiological signals are very similar. And so this kind of sleep is, if you have children, you go kiss them while they're asleep, they don't move, right? And if you kiss them here, they'll probably move a little bit. It's lighter sleep. And here's another example from primary somatosensory cortex. You cannot see much difference here. Again, slow wave sleep, you have these ripples here, and REM sleep looks a little bit like wakefulness. Ketamine xylazine anesthesia is different. Then activity goes down, and you have this brrr, then some brrr, then some brrr, then some brrr, right? It has some kind of rhythm there. Almost like there's some kind of oscillations, which, if you think about the model, if you're attached to the model, there were no oscillations in that model, right? So, in principle, so it doesn't look good for the model. If you think about avalanches, which was the signature that people were studying to approach this idea, could the system be critical in the sense? You need separation of time scales to calculate avalanches. You can find them here, but not quite. There are no clear silence here. I mean, freely behaving, right? FB is just freely behaving. So how will we define avalanches? So the way people had initially tried to solve that, ourselves included, was just kind of brute force or despair, if you want. Okay, I'm going to calculate the average interspike interval in this time series to obtain some time scale at which things are occurring. And that will be my rule of thumb to bin the data. I didn't mention that yesterday, but that's what Beggs and Planks did in their original experiments. Because, you see, I have a bunch of activity in different neurons here, and I have to calculate avalanche sizes. I need to bin the data. If I choose a bin this size, there will be no silences. It's just one giant avalanche of infinite size. Doesn't make much sense. If I choose a bin of 1 nanosecond, there will be infinite avalanches of size 1. Everything is a silence. It doesn't look reasonable. So there's this catch in this field, which does not exist for models, because in models, in a model, once activity goes away, it goes away forever, and you have silence, as much silence as you want. So that's fine for simulations, for theoretical physicists. Here you have to make more or less define what silence is. So there's this whole game about playing with the beam. I'm just telling the story in 2010, which is when the paper came out. But for the record, nowadays, people are taking this issue much more seriously, and some people work on that, playing with the beam, understanding what it means to choose and whatnot, okay? I'm not going to delve much into that, but some people are. So people just use this rule of thumb of taking the time being as the interspike interval. And this is calculated for each situation. So here, presumably, the size of the beam will be different from here, right? So you choose the appropriate time being for each different situation, and then go on, try to do this avalanche analysis and see what comes out. So if you do this for this data here, you have some time beam, which is this thing here, and you bin the data and you look closely, and you see what you get. So you have some silence here. You have an avalanche with 2 spikes, so the size here of this avalanche is 2. Another silence, another avalanche of size 1, silence, an avalanche of duration 2 and size 2, silence, avalanche of size 1, silence, avalanche of duration 3, and size 1, 2, 3, 4, etc. And you collect the statistics. By that time, people, ourselves included, were mostly interested in size distributions and were forgetting about duration distributions because they were too messy and ugly. So you do this run for one of these situations. Remember, we have 27 situations for each of the 7 animals, right? Combinations of brain region, behavior state, and stage of experiment. We didn't know what mattered or what not, so we had to look at everything. And here's one sample of a time series. Please note the log scale of avalanche size of one of these guys. So this is for awake animals. This is awake time. So the animal, rats, sleep and awake several times. So you have to put to glue together these different awake time. Most of the time it is awake in those experiments because they're excited about the objects and whatnot. So you see that you look at this time series, again, log scales. This is 1, 2, 3, this is 100. You have at least 2 orders of magnitude here to play with. So maybe those avalanche sizes are scattered along 2 orders of magnitude, and you could hope that you will find some kind of power law because they have all sizes. So you collect, yeah? Yes. Not many. They'll have less than that. Yeah. Eventually, if you increase the number of neurons sufficiently.
Well, so which interspike interval are you choosing?
Oh, so for each situation. So here we calculate for this data set the average
across all the neurons.
Across all the neurons. Yeah.
Yeah. So that means that in some sense, on average, each neuron should have 1 spike in a bit.
It's actually the inter-event interval. So we put all those neurons together in a single row, all the spikes together in a single row, and then calculate the average interspike interval. So if it's not quite short because there are not so many neurons. So this recipe fails, obviously, if you have today, with hundreds and thousands of neurons, this strategy will fail miserably. And it does even before that. With the current MEG data, we try to do with some data.
But if I understand correctly, then the average number of spikes in a bin should be 1 or something,
Uh,
or order 1, or maybe it should be 2. I mean, in some sense, you've chosen your bin so that the mean number of events in a bin is something, right?
yeah, yeah, I think it's correct. Yeah, yeah. So what we do here is we collect these statistics and make the plot, right? Again, 27 combinations. So let me show you the results, the size distribution of spike averages for the freely behaving data, not for the anesthetized animals, ketamine, xylazine, anesthetized animals. As you can see, they are perfect power laws. I'm joking. Just because when I was in my master's, doing my master's thesis, I would go to conferences during the days of self-organized criticality. People would make plots like this and pass a straight line all the time and claim that they were power laws. And if I didn't draw the fits, just put the points and just force my hand and draw a straight line, you might get fooled. The visual impression, you know, if one passes a straight line, you believe it's a straight line. But they are not straight lines, right? So they are not power laws. The lines you're seeing, the best fit for this data that we could find, at least, are log normals, log normal distributions. And probably because of the way we choose the bins, this normalization, they are incredibly similar across the sleep-wake cycle. I'm sorry, I didn't explain. So this is pre-exposure, exposure, and post-exposure, V1, S1, hippocampus, and the 3 colors are 3 different behavior states for each combination of brain region and phase of the experiment. They are rather similar, and they are not power laws. So it took us a while to understand what was going on. First possibility, well, the brain is not critical after all. You don't have a power law, period, the end. That's a simple answer. You can go with that. Or there could be another issue going on here. And there is an issue going on here that we started to explore together at the same time that another researcher in Germany called Viola Prisman was exploring, which was the idea of undersampling. The fact that differently from a local field potential, which when an electrode measures local field potential, it's measuring activity all in a large range around it. When measuring spikes, you're only looking locally around the electrode. So let me show you one attempt we had to explain this data. Okay. If we assume that criticality has some bearing on it, so that's what we did. We took a model, just like the model I showed you yesterday, but in a 2-dimensional grid, okay? So each dot here is a model neuron. We connected them to nearest neighbors. So this is like directed percolation model in 2 dimensions. And we tune the probability of connection until it's critical. We know how to do this, okay? When you do this, you calculate the probability distribution of avalanche size. It gives you a perfect power law with the different exponents, not 1.5, because it's 2 dimensions. Though it's a different universality class. It's not mean field directed percolation. It's 2D directed percolation. It's 1.28, actually, not 1.2. Okay. And this is the simulation results. We can do this. It's just a model. Now, if this were the rat's brain, this is not what we'll be measuring because we don't have that many neurons in our data set. We have way less. And the ones we have, we know come from electrode matrices whose electrodes are spaced several neurons apart each other. So I'm going to draw one of those brain regions for which Tiago had the exact coordinates. So can you see 16 electrodes here? One electrode here, one electrode here, another one, 1, 2, 3, 4, 1, 2, 3, 4. The red triangles correspond to the number of neurons that each electrode was measuring according to the data we received. So we had the identified electrodes. The spacing here between the electrodes is more or less, I don't remember, 8 or 10 body cells here, which corresponds to the actual spacing between electrodes and typical size of a cortical cell. Okay? Now, we take this model, which is critical if you measure everything here, and we just run the model again, calculating avalanche statistics, but only on the red triangles, which is what you're measuring in your data. So we're trying to model the insufficiency of the experimental apparatus. So imagine that an avalanche started here somewhere in the black dots, and then we would spread some more, somewhat fracture, like a fractal, right? Spreading like this. It's non-deterministic. It has a fractal structure, and it's making some neuron spikes, some other not. And then an avalanche is over. If you could count all the spikes in the black dots, and you collect the data, you get this thing here. But if you're looking only at the red triangles, you get way less spikes. Not only that, what is considered one avalanche here, because it spreads and then dies out, if you're looking here at only the red triangles, maybe it will start here, and you see a blip, see some spikes, and then it goes here, it doesn't get this guy, it doesn't get this guy, it will get these guys later. For you, this could be a silence in between those two, what is actually one avalanche. You're seeing it as two or more, because you just put those silences there. You have to choose some bin, remember? So there are at least two mechanisms by which you're underestimating the size of large avalanches here because of undersampling. Is that okay, what I'm saying? So what I'm going to, yeah? I think you should use the microphone, right? To ask questions.
No, just the first simple undersampling, not considering the red the red triangles, but just doing like measuring just 16 black circles, as if you have just.
Yeah. Well, not in that paper, but in a following paper, like years later, then Tiago went on to explore this possibility in excruciating details, like simulating larger and smaller and larger fictional electrode arrays and denser, and not changing the density and changing the spacing and changing whatnot and changing all the parameters. And then he explored. It came out in another paper, not here. Here we were just desperate. What do I understand? So, what happens? Yeah.
You had in a previous slide mentioned about introducing novel things in free rats. Are you also going to be talking about that?
About what?
Introducing novel things like food and new toys.
Yes, yes. Well, yes and no. I promise to come back in my conclusion, but what you're going to see here is that the way that the question that we are asking about size avalanches, the size of avalanches, etc., and distribution like this, you will not see any difference between when the animal is doing nothing or when exploring novel objects. But I'll comment on that at the end a little bit, okay? Thank you for the question, actually, so I don't forget. So here, the question is, okay, what we're doing experiments is measuring avalanches in the red triangles, if we take the model at criticality and measure only the red triangle, avalanches in these red triangles, what is the distribution that we obtain? The distribution that we obtain is this one, which then is a much better fit to the experimental data, which is in blue. So this could be, this like saves the face of criticality. Well, maybe it is. So this does not exclude the possibility that it is critical, and that you're not seeing traditional signature, which you're starting to see it's problematic. This avalanche thing is problematic because it's prone to this, in principle, to this kind of problem. And then, like I mentioned, Viola Prisman and Göttingen was someone who explored this issue of subsampling, of undersampling in neuroscience, in this kind of data, for many, many years. And I just mentioned that. So, but this kind of, you cannot exclude the possibility that it is critical. I don't know. It's from a model. Not very convincing, but okay, it's still nice that gives you this result. And you can do some analytical calculation to understand at the mean field level why it should be a log normal, because you're splitting things, and you can argue that why this gives you a log normal, okay? So this model is nice because this idea is nice, and then because it recovers the experimental data. So this solves one problem, more or less. It creates another one. Why? Which is the fact that if you look at size distribution for ketamine, xylazine, and acetase data, then you have power laws. Experimentally. Well, more or less. They're okay. Now you don't have a model for that, because these guys are subsampled in precisely the same way as freely behaving animals are. So, what on earth is going on? We don't exactly know, right? But so we have this nicer signature of a critical system for anesthetized rat, which is kind of frustrating, because if the signature only appears when the animal is doing nothing, what do we do with the original rationale for this whole idea that it will be somehow functionally useful? I hope I'm getting you confused, because we were, okay? Fine. And by the way, exponents vary, right? It's not 1.5 exactly. You have some uncertainty in this fit, which is not shown here, unfortunately. But like I mentioned, this is a kind of an anesthesia from which the animal can recover. So the darker, the darker the color, the deeper in the anesthesia they are. And I think the curves here are like half an hour apart from each other. So the animals eventually wake up from anesthesia, and as they wake up from anesthesia, these quasi, more or less, power laws get morphed back to log normals as the animals wake up. So this is the consistent. So we have the two phenomena in the same animal. You can do this just because of this type of anesthesia, right? And okay. You were not satisfied with this. Dante Chialvo joined our collaboration. We were discussing a conference. He saw the results of the log normals in Thiago's poster in some conference. I don't know, forgot which. He has ideas. This is why you should bring posters to the conferences and talk to people, and eventually some ideas get developed. It's a beautiful sociological event. We keep forgetting how important social ties are in science. They're very important. Talk to your friends, talk to people. So here I have the time series again log scale of the size of avalanches in G3 animal. one of the animals, right? So it doesn't show much. And so one of the techniques people use to find a very different kind of signature of criticality is just looking at long-range time correlations. So you have this technique, I don't know how familiar you are with it, it's 30 years old, but now it's called the trend fluctuation analysis. Let me briefly walk you through this. The idea is you have a signal here, and operation, what you do, you have some fixed window size. Inside this window size, you fit a linear, you fit a straight line here, and you calculate the residue, root mean square of the residue around this line. And then you see how this root mean square residue scale with the size of the windows. Presumably, okay, the fit here will be better than the fit here, right? Because you have a larger window, they have more fluctuations around the line, so this residue will increase as increased window size. Now, it turns out that if this residue scales with the power alpha of the window size, then you can relate this with the autocorrelation function, which if decays with time to the minus gamma, then this gamma is related to this alpha with some relation. And if you studied the Wiener-Khinchin theorem in your, where do we study this again? Which class is it? I don't know, any of them? Statistical mechanics probably, right? And then you relate this exponent gamma with the beta of this power spectrum density. So beta is 1 minus gamma, and which is 2 alpha minus 1. Essentially, it's a very straightforward way of obtaining this exponent in an easy way. So, and there are interpretations for this. So essentially, if this alpha here is like 0.5, this is essentially white noise, nothing interesting going on. No long-range time correlations. But if alpha is larger than 0.5 and 1, you have persistent long-range power law correlations, and if alpha equals only like 1 over f noise. If you put alpha 1 here, it gives you 1 over f, right? So we did this we applied this technique to this kind of time series for the different, again, 27 combinations, blah, 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 right? For each red. And what we obtained, that the free behaving data has very nice power laws here for the root mean square deviations with exponents which point to very long-range time correlations. So in the temporal domain, you do have something which is compatible in principle with a critical system, which has no characteristic time scale. Remember that this size distribution of avalanches for these freely behaving guys are problematic, like these log normals, which may be compatible with power laws if you do undersampling. And anesthesia is fine. Anesthesia gives you, it's fine for power laws. It gives some reasonable power laws. Here, the freely behaving data gives you significant exponents pointing to this temporal fingerprint of criticality, if you want. And then we went to look for the anesthesia data, right? Let's look at the temporal signature of criticality for anesthesia data. It's not even a straight line here. So the anesthesia data, which gives you reasonable, for this number of electrodes, reasonable power laws inside distributions, gives you a lousy response in the time domain. And if you're confused, welcome. So were we, I think, so which one is it? Right? Maybe they're just different things. Different signatures mean different stuff. So you don't get nothing, you get nothing here in anesthesia. Some people, so we were, puzzled by these results. And this is very consistent across animals, okay? This is, if you want pre-exposure, if you want, again, but you do this for whichever brain region and whatnot, it kind of works. So, in a way, this is nice because, remember, we had a few electrodes, like 32 electrodes, but then you have to insert part in V1, part in S1, part in hippocampus, different brain regions. So we don't have many electrodes per region. It's problematic to go look for power laws and size distributions if the size of your avalanche cannot vary much. So going to time domain, it's interesting because, well, time you have as much as you want, as much as you can keep the experiment running, or right? But again, this was not our experiment. It was an old experiment, which was not done for these purposes. So we're going back to this time series, and this was Dante Calvo's idea. Well, why don't we have a look at waiting time distribution, which is what people, for instance, in earthquake literature do by studying how long you have to wait for another earthquake. People have obtained all kinds of interesting results in that other kind of literature. So this is a little bit tricky. And what we're doing here, again, is inspired in this earthquake literature. You notice here that I drew this dashed line with some size SC, right? What I'm going to look at is the following. Let's assume I establish some threshold, which then I'm going to vary in a minute. And I'm going to ask, what is the probability density of waiting for an avalanche of size at least SC during interval tau? It's probability density of a given time interval. So the question I'm asking, okay, suppose you are swimming in the sea. A wave comes. And then I ask, well, how much do I have to wait for another wave? My answer is, well, it depends on the size. If it's a 1 millimeter wave, another one just came around. And another one, and another one, and another one. Well, that's not a wave. Well, what is a wave to you? Like this? Is that a wave? Oh, another one just came. Well, it's like 2 meters. Okay, then wait. Right? So that's what we're doing here. We're calculating the probability density of waiting for an avalanche of size, at least SC, so whatever comes above this guy during interval tau. You can do this. This is what you obtain for different values of SC. So for an avalanche of size 1, the waiting times are around here. There's a red curve. Okay? Please note the log scale here in the horizontal axis. The larger the size SC, the longer the average of the curve here, right? So for avalanche of size 1, which is the smallest possible size of an avalanche, it's like one spike, I mean, the average waiting time is somewhere here. I don't know, a couple of milliseconds. For avalanches of size at least 2, this orange guy here, I don't know, I'm just guessing here the number, okay? I'm just doing a straight line but visually. The average waiting time was probably like here, 15. For average of size 3, you're probably here. This is this yellowish whatever curve here. For average of size 5, it's probably here, and you get the picture. Right? The larger the avalanche, the longer the average waiting time. Yeah. So what? I mean, just makes sense. It tells you nothing, in principle, at least, right? That's reasonable, right? That it should go like this way. The longer, the higher your bar, the longer it takes for this thing to happen. So, so in principle, if you have two parameters here, you're describing what happens from the scale of 1 spike and a couple of milliseconds to 36 spikes and more, because everything's above it, and things that are happening on the scales of thousands of milliseconds here. That's the time you have to wait for a very, very large avalanche to come. So, in principle, you're describing things which are of different scales. Short time scales, small things, longer time scales, large things. You have two parameters. Now, this is time. This is probability density of a given time. So it has the scale of D. Dimensional analysis is time to the -1, right? Because if you integrate D tau, it doesn't matter this thing, D tau equals 1, right? So this has dimension of tau to the -1. So you take this time here, this average time for each, say, the red curve has a dimension of time. You divide time by this guy, so you have a normalized time. Okay? And here you multiply this average time by this D for the corresponding, each corresponding curve for each different color for its appropriate average time. Okay? And you redo the plot and see what happens. I promise you it's very nice. So, if you do this, this is what you get. You get a very nice collapse of the curves. So what is written here is that so this guy times the average waiting time, and this guy divided by this guy can be described by a single function. So this guy times this guy is a single function of this guy here. And this is scaling. What this means is that the recurrence of avalanches of different sizes as C, from 1 to things larger than 36, is governed by a single function. I have one function to describe things in all different scales, from small avalanches to large avalanches, from a few milliseconds to 1,000 milliseconds here. Right? And this is one of the signatures that critical systems are also expected to have. And now, you repeat this thing for all those 27 combinations for each of the 7 rats, and you get this scaling in essentially all cases. Here in gray, I'm showing the non-normalized things, so different. So this original density, probably density of waiting time here, as a function of time in milliseconds in gray, and in the colors, I have the normalized version of D and the normalized version of T. This is one animal 5, animal 4, animal 1. This is during waking in the hippocampus, during exposure to novel objects. During slow FB4 in slow wave sleep, V1 pre-exposure, FB1 REM sleep, S1 post-exposure. It doesn't seem to matter much. They all seem to fall into this nice lines, nice data collapse. what do you think will happen in ketamine, xylazine anesthesia? Would you take a guess? No? We had a hint already, right? What I showed before was that if you look at long-range temporal correlations, freely behaving data shows it, and anesthesia data does not. So what would you expect if I try the same kind of combination here? Well, look, there's some luck here involved, okay? We just happen to combine these things, multiplying here and dividing here, and it just worked. It didn't have any obligation. It just did. I don't know if it had an obligation or not, but it did work. So if I try the same approach for anesthesia data, you don't have a data collapse any longer. I mean, at least for this combination, it doesn't work. Different anesthetized animals, the V1, S1, they didn't do hippocampus work, but I doubt it, okay? So something else is going on here. This is the same data. This is the same data that gives you these nice power law size distributions. And this is the same data which gives you nasty, ugly log normals. I mean, not ugly, but not power laws that you wanted, I don't know. So it's very confusing, right? It's as though freely behaving data is giving you some kind of signature of criticality in the temporal domain. In the spatial domain, you don't know, maybe it is, but you cannot find it because of subsampling, of undersampling. And because you're using this signature of criticality, which is avalanche size distribution, which I'm trying to convince you, and I'll convince you tomorrow, probably even more, that is problematic. Which is why we were in so desperate need of other theoretical techniques to address this data, which is what Bill's going to talk about, I don't know when. Today. Okay. So he'll come to rescue, okay? And on Friday, I'll talk about what happens if you take his techniques to this kind of data. Not this one, but. Okay, so we have this data collapse here. Notice that, like, I talked about this before, this is a dimensionless quantity, and so is the argument of F here. They're both dimensionless quantities. So these normalized distributions of different animals could be compared in principle. I don't know. Can I? It would be nice because then I'll have a clear picture of what that function looks like, right? I cannot compare things from different animals if they have, well, I will have a large animal, a small animal. Well, the different sizes or whatnot. Well, I cannot compare masses. That would make it much sense. But here, this thing are dimensionless. I would like to compare them. And if I take freely behaving data, this is non-normalized, from all these animals, and I renormalize all of them and put them all together here, now the shape of the scaling function gets much better. Right? It gets much better. Like this, you have this, I'll comment on that in a minute. This fit here is a double power law. And a double power law is precisely one of the functions that they use to fit waiting time distribution for earthquakes, another system believed to be self-organized critical, in the sense that they give these power law distributions of earthquakes. And Just like in earthquakes, you have this on short time scales. Why the quotes? Well, short. Short as compared to what? Well, it's a normalized time. Short as compared to the characteristic time of avalanches of that size. Remember, you choose your size, avalanche size, minimum avalanche size. Then this gives you an average waiting time by which you normalize time. So in the short time scales, you have this what will be called Omori's law, or the reflection of Omori's law, original Omori's law in the waiting time distribution, which is a small exponent, meaning that you have aftershocks after a large earthquake. Another one will likely come. So you have these reflecting correlations among the next one and the next one. Whereas for long time scales, again, long. Long meaning long for that size of avalanche after this crossover region here. Then you have this large exponent, meaning that's almost memoryless, almost Poisson-like if you want, right? I mean, if you wait long enough, you lose the correlations. It doesn't matter, right? But how much you have to wait? Well, it depends on the size of the avalanche, right? So you can do this plot for each experimental condition of the 27, 3 brain regions, 3 behavioral states, 3 phases of the experiment, and calculate these exponents alpha and beta. I'm sorry, we are again, this is not the alpha of DFA, okay? This is this alpha here. You calculate this pair of exponents for each of these conditions, and they are relatively stable. So there's a kind of quote-unquote universality in the sense that exponents are more or less stable no matter what you do. I'm not showing you these results, but as always in biology, you have to have some control. So if you shuffle the data, then these exponents get more trivial, and they get spread all over the place, and you lose this kind of quote universality, this constancy of these exponents here. Okay? So let's pause to appreciate that we have a single scaling function describing 7 rats over about 5, almost 6 decades of data. And given all the differences, I think this is nice. Questions so far? I'm not quite done yet, but just questions. It's a little bit too much, perhaps. I told you it was going to be messy, right? Right, so this is my favorite results of this paper, personally. And there's also that, long range time correlations that you observe for the freely behaving data. I'm not mentioning the results for anesthesia, which, as we saw, are the opposite, right? In the time domain, nothing happens here for anesthesia data, and nothing happens here. On the other hand, for size distributions, anesthesia is just wonderful. Ketamine cells in anesthesia. So which one is it? So this is free behaving rats. Let me go back to where we were yesterday. I showed you that very nice model. I mean, I like the model. because it gives you some nice results, which is this one, right? If you have low enough coupling, your activity will reach this absorbing state. If you have high enough coupling, it will go to an active state. And if you have a critical point, it will reach an absorbing state, but it takes longer. It doesn't have a characteristic time, etc. Right? Let's try to imagine the power of this model in addressing these issues here. Well, it's an active absorbing phase transition. If I'm criticality in any direct upregulation-like model, dynamics sees once the absorbing state is reached. That's what this class is all about, right? So if you ever work with that kind of model, my students know if you have to simulate, how do you simulate this model? Well, you code for the topology, for the neurodynamics, and then if you start if you want to do avalanche analysis, you don't start it like this. You start with everyone quiet down, so everybody's in silence. Nothing happens. All the neurons at 0. Then you pick one at random and spike it, flip it, and watch this activity spread until it dies out. And then you count the number of spikes. That's in one avalanche. It stops. You have to keep monitoring on your code. Okay, when it stops, an avalanche ended. You want another one? Well, we start another one. Pick another neuron and flip it. And then, you have another avalanche until it dies again. Right? So, separation of time scales in the model is imposed by hand. It just happens. It's just there. You don't have these long-range time correlations in avalanche sizes because they are, by nature of the model, in the simple model, they're independent. There's no memory in the system what just happened. Therefore, this will not happen. Even more, obviously perhaps, is the waiting time distribution. Well, I just told you, if you simulate the model, it will reach the absorbing state and stops. Well, when will the next avalanche come? Well, whenever you code it, it will come. I don't know, right? It will not emerge spontaneously. In the model, it's just you code it, and then another one comes. Typically, you wait nothing. Code it, right? So there's no way this will come out. Not any simple model, just like the one you showed. Well, maybe if you do something more more complicated with different ingredients, with some memory and some whatnot, well, maybe it will. But yeah, be my guest. I mean, so to be clear, I'm not aware of biologically plausible or reasonable models which can reproduce these results so far. Okay? If you want to try, I would love to see. Not because it's impossible, because we have not done it. So maybe someone has it, didn't see, but I don't think so. Yes?
If you do the simulation for the very big, a lot of neurons, and then you downsampling and use just the downsampling, your avalanche, like the activity of all the network will be still active, but you are going to think that it's an avalanche. And then, I mean, the next avalanche will appear even you have to...
It could be. Just because you could have the blackboard could be a supercritical system. I'm just looking here, some activity goes there, and then it comes back. I didn't see it working. Perhaps. Okay, so I will not have time to talk about this, but there is a model that goes in that direction, not the way you're saying, but it's called the cross model, the critical oscillation model, which was proposed by Klaus Linke-Hansen in Amsterdam. So what he has is a network with excitatory and inhibitory neurons. They are connected to each other in a square lattice, and they have a very small input. If you have, which keeps everything more or less functioning all the time, so no real absorbing state. And the connections here and the time scales involved and the nature of the model are such that if you play with the excitatory connections and the inhibitory connections, you reach a line below which you have some kind of near silence, not real silence because you're fueling these neurons something, and here you have self-sustaining activity. But, so they have activity, and they have time, and they have something like this. If you are here crossing this threshold, presumably a phase transition, but it's hard to tell because the model is very complicated, has many parameters, and it's not obvious, many control parameters, I'm sorry, and it's not obvious what a control parameter for this case would be. When you're going from here to here, you start seeing this activity trying to go up. So trying to raise from some kind of near absorbing or not quite absorbing state into something which is active, but also in a power spectrum, as you go from 1 to 3, going through 2, you have like for 1, something not interesting like this, and when you are at 3, you have a peak in the power spectrum, and you are, this is 3, and you are at 2, the peaks is starting to emerge like this. So it looks like a
Did you use this model with Leonardo Dalla Porta?
yeah, so this is what I'm describing is the cross model. It was published by Poyle collaborators in Journal of Neuroscience 2012, I think. And I was fascinated by the result. Why? Because the way they define avalanche is like this. They put a threshold. And whenever things are above the threshold, you have an avalanche, right? There's an avalanche, there's an avalanche, there's another avalanche. And now you saw, it's something like what you were suggesting. Now, since you don't have a true absorbing state, this thing will emerge due to the dynamics of the network after this thing has happened. So there is a possibility of correlation between consecutive avalanches. So this model, you can marvel, has here power law distributions of avalanche sizes, and this f of n of DFA, long range time correlations, at the same time, at the same point. We could say, well, this solved the problem. Well, it didn't solve for me because let's go into the details then. This is a two-dimensional network. The exponent here for avalanche is 1.5, which is a mean field value. How come do you get a mean field value for a two-dimensional model? I couldn't get my head wrapped around this thing. So what Fernando is saying with Leonardo Dalla Porta in PLOS computational biology, it took us a long time. 2019, 7 years after that, we revisited the model, simulated larger models, and tried to understand how on earth this exponent tau could be 1.5 in a two-dimensional network. And it turns out that this exponent tau, as well as the exponent tau t for duration distribution, they all depend more or less continuously on this threshold here. Which is another nightmare, because if you go to your phase transitions 101 class, you learn about universality classes, you know that you have a phase transition. What is it characterized by? Well, well-defined exponents. If my exponents in the model depend on whatever I do here, well, good luck with that. Right? And actually, this dependence of exponents on stuff, on things you do, either to your data or to your model, is something I'll go back to tomorrow, actually, in order to, again, show you the problems with this kind of measurement. Did I answer your question? Yes? So, is mean field calculation of satisfactory model for supposedly critical brain dynamics? Well, the pros I had listed late at the end of my lecture yesterday and today, that has these nice things here, but it fails here miserably in more complicated scenarios. So maybe it's useful in simple experiments, but less so in more interesting setups. To wrap up, let me answer your question about the food and stuff. So, like I said, you wouldn't see any difference here, right? Even though the activity the rat is performing is completely different. It gets crazy when it's subjected to novel objects. They're very curious, they explore a lot. So, in a different paper, which is even, in my opinion, more complicated to explain, to give a talk, we explore something different. Let's look not at the distribution avalanches, but let's look at the identity of the avalanches. So you have these size distributions being preserved more or less across conditions, but is the animal changed after it is exposed to novel objects? You think, well, yeah, I mean, I guess so, right? But is this reflected in the avalanches? Not in the distribution of sizes, but who cares about size distributions? So when you look at the identity of the avalanche, please note the computational tour de force that this involves. This is Tiago's merit again. So you have an avalanche which is neuron 1, neuron 5, neuron 17, 20, and then stops. And then another one avalanche has a different spatial temporal sequence. And then another one, and then another one, another one. So what you do is look at this data and see, okay, is there some kind of consistency here? Some repeated patterns. So yes, so there are families of avalanches, some kind of repertoire of avalanches, which is more or less stable during pre-exposure. During exposure to novel objects, even though the size distributions are the same, these log normals, the identities of the avalanches shift and change. You have new families being created. And then these remain later. And then this whole issue of how this depends on sleep and not, which is connects with Siddhartha Ribeiro's interests. So it's a completely different paper. So we looked at it, right? And it's very nice result, I think, also very complicated to explain. But so yes, contact with novel objects change the way, the identity of the avalanches, right? I can give you the reference if you want. So again, as a sociology of science, as someone coming from models and theoretical physics, and seeing the model I was attached to fail miserably here, and at the same time, very interesting data, results coming from data, I understood I had to make a decision in my life. This is 2010. I had a permanent position for about 8 years or so, still young. And I understood that dealing with real data was something that enriches my life, the scientific results and scientific questions, even though I cannot answer them. So I was inspired by two people that I admire very much. You know them both, Bill. One is my good friend Gabriel Mindli, who originally as a theoretician decided to set up a lab with birds in the physics department in Buenos Aires. And he does, together with Ana Amador and their fabulous students, very interesting experimental work informed by theory in a way that I find extremely elegant. And it's hard to talk about him because he's a good friend of mine as well. But I'm a big fan of his work. So, and the other one is Holland Copelli. Holland Copelli is a by now retired professor of physics in São Carlos. Field theorist, theoretical physics for most of his scientific life, and then later in life, the second half of his career, after spending some time in the US, decided to set up a electrophysiology lab in São Carlos with flies and electrodes and stuff like this. So I decided to take a similar plunge after this behavior. I'm still good friends with Siddhartha Ribeiro. We still collaborate, but I thought it was time. I still had time to try to set up an electrophysiology lab in Recife, where I am. I'm not an experimentalist. I don't do surgery. I cannot do this thing. But I convinced someone who could. So Pedro Carelli is another PI in our lab. I talked to him during one of those Society for Neuroscience meetings in the US. He was in France. He came to Recife, become faculty, and together we assembled a lab. Now we have Nivaldo Vasconcelos, whom you also know, whom we recruited back from Portugal. He's now a professor in engineering. And we set up a lab which very slowly was assembled under less than suboptimal conditions that Brazilian science can offer outside of São Paulo. And the results I'm going to show you tomorrow are already from this second part of my life, in which even though I don't do experiments, I'm there trying to help put the lab. And going back to these questions almost 10 years later, that's the time it took for the first results to come out. Okay? Thank you.
I have a question about the model. I think I lost the idea of the model. Okay, so do you use experimental data as an input in your model?
No.
Okay, that was my question.
How would I, what do you mean?
Because I was working with this generalized Ising model, and they use like a connection matrix to try to have to get the correlations of the rest state of the brain. And I was thinking that maybe in your model you use that, but I think I missed the idea of the model.
No, the model is much simpler. If you want to look at the data and from that produce a model that spits out something that looks like the data, I recommend you go to Bill's paper, 2006. Schneiderman, right, the first author? It's a Nature paper in which he uses maximum entropy, which he mentioned in his first lecture. In our group, we have a folder of must-read papers. Don't talk to me if you haven't read those papers. You have to read it because they're... So more is different is there, Phil Anderson's paper. And, well, you have to read these Bags and Plants if you want to talk about criticality. And Bill's paper is there. Not only this one, other ones, but this one, highly recommended. You're going to talk about maximum entropy? Okay. Yeah, great. Did I answer your question?
Yeah.
Sorry, yeah. Thank you.
Other questions?
So in the double power law distribution that we have seen in the waiting time, is there a model that explains where this bend will be? The kink in the...
I don't know of any neuroscience-like model that can do this. Maybe there probably are models in earthquake literature for earthquakes, and I bet you can find someone that will take an earthquake model and tell you it's a brain, but not. I mean, I don't know. Leonardo Dalla Porta tried to obtain those results with the cross model, since it has these long range time correlations. I think he didn't find it.
Okay, thank you.
So the window is open for anyone to try. Look, it's not like we tried much either. We didn't. We have other things to do, but to the best of my knowledge, it's an open problem. It hasn't been solved.
I was thinking if there will be some phenomenological significance of these two parts of the distribution. in behavior.
Well, the interpretation is, again, comes from earthquake science, right? This part here is characterized by this, in earthquake literature, the dynamic of aftershocks. So after a big one, will likely come another one, and so the waiting time does not decay fast enough. That's what the small exponent means, right? Whereas for long times, again, long compared to some normalized time which scales with size, then it becomes like decorrelated. So this is the interpretation of the double power law, as far as I can see, yeah.
This is a question I have often when I see these experiments with these mice that have the electrodes in their brains. How well is it understood how much the experience of having their skulls opened and electrodes inserted in their brains affect their behavior? Like, are these mice in distress while they're doing their activities? Are they in more pain than a natural mouse? Like, how much would this might affect the data of their neural activity? That the fact that
Again, put myself in Bill's position like yesterday. I just appreciate the efforts of my experimentalist colleagues who do this thing. So my best answer, anyone can help me.
And just be clear, not because I necessarily want you to talk about the mice's welfare. I'm interested also in the way that, you know, a mouse in some kind of body distress may not have typical neural activity, for instance.
So my fully honest question to zero order perturbation theory, I don't know. So the first order perturbation theory, what I would guess that those guys had probably thought about this thing, and I think they care about this thing, and they try to make it as painless and as respectfully as they can. And I don't know to which extent this has, you could have an answer to that. How would you know? I don't know. But I think they try. I mean, for behavioral tasks, with and without the implant, how will they behave? So I think for behavior, I mean, you can try something and you do before the implant and after the implant, the results are more or less comparable, for instance. But so the animal is doing something, because it's, when there's a task, there's motivation. Does that mean that they don't feel it or they don't care, the animals? I mean, I'm not sure, but I don't know. And so you have all these techniques. I mean, these are rats, by the way, they're not mice. It's very different. And you have this thing now with the head fix, right? That Bill showed yesterday. I'm going to show you also on Friday, because now we can explore awake behavior while implanting a measure of thousands of neurons. It probably doesn't feel good to have your head fixed, but they're paying these mice very well with money and juice and stuff like this, and excellent health care. So I don't know. We don't know. But I think people try to test that, and very insufficient answer. I'm sorry. I don't know.
Yeah. Hi. Here.
Yeah, sorry. Yeah.
So I wanted to ask you. Well, you mentioned that the fact that this recurrence of avalanches could be like governed by a single function is something you will expect from a critical system. I would like to ask you, like, why will you expect that from a critical system, and if it's maybe the same criticality that we talked about in last lecture, like this branching ratio of 1, or is criticality understood in a different way?
Yeah, it's probably criticality understood in a different way because we're not sure branching ratio equals 1 is like the branching model. It's just a model which explains some data. It does explain that. I don't know how it would explain that. Maybe it does, and I don't know how to do it. So like here I get some other signature. This kind of thing here is that what you're going to obtain when you study critical phenomena in textbooks in the beginning. So it depends on the temperature, it depends on the magnetic field and whatnot, and at a critical point, you manage to combine these different dependencies with the smaller number. It doesn't depend on all of them separately, but together according to some scaling law like this, right? So free energy and stuff like this. So that's where the comparison comes from. And this is, I mean, I'm talking about regular equilibrium statistical mechanics and textbook stuff, but here, this particular shape here, for weight and time distributions, is something that they see in earthquakes. And that's why. Okay?
More questions? If not, we should go to the coffee break and come back to Bill's class.
Thank you.